Hello dear students, welcome to this program. Today we are discussing an important topic, that is Introduction to Enzymes Part 2. Before discussing in detail Introduction to Enzymes, let's first know its objectives. Introduction about enzymes, multi-enzyme complexes, cooperativity, Zymogens, Isozymes, Abzymes and ribozymes. Let's begin with introduction about enzymes. Enzymes are macromolecular biological catalysts. Enzymes accelerate or catalyze chemical reactions. For example, catalase. It catalyzes the decomposition of hydrogen peroxide in living tissue. One catalase molecule can convert 5 million molecules of hydrogen peroxide to water and oxygen each minute. Almost all metabolic processes in the cell need enzymes in order to occur at rates fast enough to sustain life. The molecules at the beginning of the process are called substrates, and enzyme converts these into different molecules called products. The most striking characteristics of enzymes are their catalytic power and specificity. Catalysis takes place at a particular site on the enzyme called the active site. Nearly all known enzymes are proteins. Proteins themselves are formed of amino acids. However, proteins do not have an absolute monopoly on catalysis. The discovery of catalytically active RNA molecules known as ribozymes provides compelling evidence that RNA was an early biocatalyst. An enzyme works by lowering the activation energy, which is the amount of energy required for reactants to form products. They also lower the kinetic barrier, which is needed to drive a reaction forward and backward. A certain amount of energy contained in the molecules is needed when the two molecules react together to form a product. If the two molecules do not have enough energy to react, then no product is produced. By lowering the activation energy, an enzyme allows the molecules to gain sufficient energy to overcome the barrier and form products. Now let's talk about multi-enzyme complex. Multi-enzyme complex is defined as stable assembly of more than one enzyme, generally involved in sequential catalytic transformations. Multi-enzyme complexes enhance the rate of reaction by preventing the dilution of metabolite. If we consider a metabolic pathway, the product of one reaction is the substrate for next. Such direct transfer of metabolite from one enzyme to another would avoid dilution of metabolite in bulk aqueous environment. This would in turn increase the rate of reaction. An example of multi-enzyme complexes is pyruvate dehydrogenase. It is a complex of 3 different enzymes that collectively catalyze the oxidation of pyruvate. These enzymes are lipoamide reductase transacetylase, which is a trimer, dihydrolipoyl dehydrogenase, which is a dimer, and pyruvate decarboxylase, which is also a dimer. Now let's talk about cooperativity. Cooperativity is a phenomenon in which the shape of one subunit of an enzyme is altered by substrate or some other molecule so as to change the shape of neighboring subunit. The result is that the binding of second substrate molecule to the second subunit of the enzyme differs in strength from that of the first, the third from the second, and so on. For an enzyme that shows a cooperative behavior, the relation between the two shows a sigmoidal curve instead of Michaels and Manton behavior. Now let's talk about types of cooperativity. There are two main types of cooperativity. First, positive cooperativity, and second, negative cooperativity. Now let's talk about positive cooperativity. If the change in the shape of the first subunit makes easier the binding of substrate to the second subunit, the effect is called positive cooperativity. This behavior is seen on the binding of oxygen to hemoglobin to form oxyhemoglobin. Hemoglobin is made up of 4 subunits, 2 alpha and 2 beta. They come together to form a tetramer, each subunit having its own active site to bind oxygen. This active site contains a porphyrin ring structure with an iron atom in the center. When the subunit is not bound to an oxygen, it is considered to be in tense state or T state. When hemoglobin is bound to oxygen, it is called R state or relaxed state. The oxyhemoglobin or the T state has low affinity for oxygen. When one molecule binds to a single heme, the oxygen affinity increases, which allows the incoming molecules to bind more easily. Now let's talk about negative cooperativity. In negative cooperativity, there is decrease in the binding affinity once one of the sites is bound. For example, the relationship between glyceraldehyde 3-phosphate and the enzyme glyceraldehyde 3-phosphate dehydrogenase. This is a clear example of negative cooperativity. Now let's talk about isozymes. Isozymes, also known as isoenzymes, are homologous enzymes that catalyze the same reaction but differ in structure. The differences in the isozymes allow them to regulate the same reaction at different places in the species. In particular, they differ in amino acid sequences. They display different kinetic parameters as well as regulatory properties. For example, isozymes have different Km and Vmax values and can be distinguished from one another by biochemical properties such as electrophoretic mobility. Isozymes are encoded by different genes and expressed in distinct organelle or at distinct stage of development. Now let's talk about application of isozymes. Isozymes in general can be used to meet the metabolic needs of different tissues and developmental states. An example of an enzyme with different isozymes is lactate dehydrogenase. This enzyme is used to catalyze the synthesis of glucose in anaerobic metabolism of glucose. The isozymes of this enzyme are divided into 2 forms, the H isozyme and the M isozyme. The H isozyme is expressed more in heart, whereas the M isozyme is expressed more frequently in the skeletal muscle. Both isozymes have 2 polypeptide chains. Each isozyme shares 75% of amino acid sequence for the chains. Both isozymes metabolize glucose, but the difference is that H isozymes have a higher affinity for their substrates than the M isozyme does. Another difference is that H isozyme functions better in aerobic conditions such as heart, whereas the M isozyme functions better in anaerobic conditions such as muscle. Let's now talk about Zymogen. A zymogen, also denoted as proenzyme, is a group of proteins that can also be described as an inactive enzyme. Since it is an inactive precursor, it does not hold any catalytic activity. These zymogens can be activated by chemical processes such as cleaving, hydrolysis, along with other biochemical chains. These biochemical processes often occur in lysosome, where cleavage reveals the active site. Exposure of active site allows the enzyme to become active and function to catalyze the reaction. The reason for cells to secrete inactive enzymes is to prevent unwanted destruction of cellular proteins. It's only when the conditions are right that zymogens become activated into enzymes. Following examples of zymogens will explain how zymogens are activated. Talking about pepsinogen. Pepsinogen is an inactive precursor form of pepsin. It is secreted by chief cells in the stomach. Pepsinogen is activated by hydrochloric acid secreted by the parietal cells. Once pepsinogen becomes pepsin, it is responsible for the breakdown of food. The difference between pepsinogen and pepsin is that its primary structure has an additional 44 amino acids. Now talking about chymotrypsinogen. This is another example of zymogen. It is an inactive form of chymotrypsin. It is cleaved by trypsin. Chymotrypsin is found in digestive system of mammals as well as other organisms. It works by cleaving peptides at the carboxyl end of aromatic amino acids. These aromatic amino acids are tryptophan, tyrosine, and phenylalanine. Now let's talk about Abzymes. Antibodies and enzymes share the ability to bind with compounds with great specificity and high affinity. This property has been exploited in the development of antibodies with catalytic activity. In 1986, the first monoclonal catalytic antibody termed as Abzymes was obtained. Abzymes are catalytic antibodies having structural similarity with the transition state of enzyme-catalyzed reaction. They bind strongly to the transition state with high association constant, thus enhancing the reaction rate. Abzymes are usually artificial constructs. They can also be obtained from human and animal serum. These are capable of hydrolyzing proteins, DNA, RNA, polysaccharides, and so on. Some other reactions catalyzed by Abzymes include amide hydrolysis, transesterification, photocleavage, oxidation, cyclization, reduction of diketone, and hydrolysis of enol ethers. Now let's talk about production of Abzymes. The production technique involves the following principle. The generation of Abzymes is based on transition state theory of catalysis. As we know that enzymes work by stabilizing the transient form of molecule known as transition state. If a mimic of the transition state of molecule could be generated, then an antibody that would tightly bind that mimic should be able to catalyze the reaction of original molecule. Therefore, by designing a good transition state mimic, that is, transition state analog, novel catalysts can be created. The process of making Abzymes goes like this. Step first, the transition state of specific enzyme reaction is studied. Step second, suitable transition state analogues, that is, TSA or haptens that mimic the transition state are created. Step third, introducing these haptens into organism and let immune system do the rest. And step fourth, finally, the antibodies are raised against the TSA and are recovered. At last, now let's talk about ribozymes. A ribozyme is an RNA enzyme that catalyzes a chemical reaction. The term ribozyme was first introduced by Keller-Krugel and co-workers in 1982. The ribozyme catalyzes specific reactions in a similar way to that of protein enzymes. Ribozymes are found in ribosome where they join amino acids together to form protein chains. Ribozymes also play a role in other vital reactions such as RNA splicing, transfer RNA biosynthesis, and viral replication. Some of the known naturally occurring ribozyme classes include group first self-splicing intron, group second self-splicing intron, hairpin ribozyme, hammerhead ribozyme, HDV ribozyme, RNase B, and twister ribozyme. The first ribozyme was discovered in early 1980s. This led to researchers demonstrating that RNA functions both as genetic material and as well as biological catalyst. This contributed to the worldwide hypothesis that RNA may have played a role in the evolution of self-replicating systems. This is referred to as RNA World Hypothesis. Ribozymes have been extensively investigated by researchers to try and determine their exact structure and function. Scientists have developed synthetic ribozymes in the labs that are able to catalyze their own synthesis under specific conditions. Ribozymes also play an important role in gene research and functional genomics. This was all about today's topic. Hope you understood. Thank you.